Good afternoon, everyone. Thanks so much for coming. I'm Nan Crowder, and I'm the Dean of the College of Health and Human Development. And on behalf of our faculty, students, staff, and alumni, I'm just delighted to welcome you to the Distinguished Alumni Speaker Series. The Distinguished Alumni Speaker Series is the brainchild of the College of Health and Human Development's Alumni Society. Back in 2005, the Board of Directors of the Alumni Society established the Speaker Series in order to celebrate alumni who have been very successful in their professional lives, and to allow these alumni to share their knowledge and expertise, not only with students from the College of Health and Human Development, but with the entire university community. The first lecture in the series occurred 7 years ago when Wendy Owen spoke about her role in communicating with the media and the public about the Shuttle Columbia tragedy. Other speakers include Olympic bronze medalist Mary Ellen Clark, Mario Lafortune, who's the head of Nike's Sport Research Lab, brain injury advocate Joanne Finnegan, who's an RPTM alumna, voice disorders expert Bob Hillman, a CSD alumnus, Jill Jane, who is the country's only, quote, rock star nutritionist, who combines nutrition education with rock songs for kids, and Patty Hilkirk, founder of a camp for HIV-positive children. I'd like to thank the Alumni Society for its vision in establishing this speaker series and for really continually striving to create opportunities for our faculty and students to benefit from our incredible alumni network, which is now more than 60,000 alumni strong. So I'd now like to invite Jeff Sarabach, who is president of the HHD Alumni Society and an HPA grad, if you're curious, to come to the podium to introduce this afternoon's speaker. So Jeff?
Thank you, Nan. It's my pleasure this afternoon to introduce our speaker, Dr. Mark McCamish. Mark is at the forefront of something that one of our HHD faculty recently called paradigm-shifting research. As global head of biopharmaceutical and oncology injectable development for Sandoz International, Mark leads research and development of all biologics. Biologics represent a wide array of products produced at the leading edge of biomedical research and often treat conditions for which there are few treatments available, such as rheumatoid arthritis. One of Mark's colleagues explained that Sandoz is a pioneer in the development of biosimilar monoclonal antibodies, for which Mark almost single-handedly developed clinical approaches that allow these products to be developed in a rigorous and cost-efficient way. He has also been a key figure in shaping the regulation of biosimilars worldwide and explaining these products to regulatory agencies, physicians, and the public. Dr. McCamish?
Thanks, Jeff. So it's my pleasure to be here today with you. I only have about 20 slides. I'm going to not put you to sleep, I hope. If you have questions, feel free to jump up or wave or whatever as I'm going through. The whole point is to really educate you about what we're doing, see how some of the technology that you're actually using applies, and then give you an overall view of what we're trying to do for the public here in this situation. So I hope by the time I get done, you can understand what a biosimilar is, what a biologic drug is, and the need for patients, and how we're trying to address that patient need. So I'll talk about these 4 categories: why biosimilars, what are biologics, biosimilar clinical development, and then our opportunity to impact patients' lives. First, I just wanted to share with you our business unit vision, and it's to dramatically increase patient access to life-enhancing biopharmaceuticals and oncology injectable. What does that mean? The biologic drugs that we're producing, they're very expensive, and I'll go through this in a few minutes. So these are drugs that are produced in cell models. So they're produced in a biologic system. very complex. They were created using recombinant technologies dating back to the 1980s. They are very expensive. There's about $100 billion of biologics used currently that will go off of patent between now and 2020. Our goal is to create copies of these biologics, called biosimilars, so that we can bring them out at a lower cost and more patients can benefit from these treatments, which, as Jeff mentioned, the biologics allow you to target specific cells or specific mechanisms that are not possible to be treated with the typical small molecule pills that you take. And this pathway is quite unique. Sorry. Not sure why. So it's challenging because you're setting a new paradigm. You're creating something new for regulatory authorities. You're creating something new for patients, for physicians. There's a lot of knowledge base that's there. It's a long road. It can be windy. It can have various barriers in it. So I'll tell you a little bit about this. And I guess I won't use that one. So, as you know, biologics have revolutionized modern medicine. These products are targeting receptors and cells and inflammatory cytokines that you just can't target with pills. And it starts back with understanding the DNA molecule that was decoded in the 50s. Then we understood what that code stood for in the 60s. We enabled various biotechnology in the 70s. And then we began to commercialize this in the 80s, where we used recombinant technology. So you're taking the DNA code, you're inserting this DNA code into a host cell. That host cell is tricked into making the protein coded by that DNA code, and then you can isolate that protein and use it as a drug. And some of the first drugs were erythropoietin, growth hormone, insulin, others. And in the 90s, it really escalated, and you can see all of the different drugs that are there. And these are the drugs that will become off patent between now and 2020. And to give you an idea of the cost of these drugs, the first one is Enbrel, second one is Remicade, and then there's another one, Humira. All three of those biologics are anti-TNFs. So they're injected, and they're monoclonal antibodies or fusion proteins that attach to tumor necrosis factor. TNF, or tumor necrosis factor, is secreted in the midst of inflammation as well as in cancer. And that inflammation causes this cascade of ongoing inflammation and damage in rheumatoid arthritis patients, patients with psoriatic arthritis, patients with inflammatory bowel disease, etc. So by designing a monoclonal antibody to capture TNF, you decrease that inflammatory signal and treat the patients. And they have a dramatic response to TNF. So you can take a rheumatoid arthritis patient, and you've seen these patients that develop joint abnormalities with the ulnar deviation and difficulty in using the joints that are there. All of their joints are impacted and are eventually destroyed. If you treat patients with these anti-TNFs, you prevent that joint destruction. You essentially make them feel normal, which is an amazing thing that you can do. The problem is, each of those three drugs together, they sell about $25 billion a year. And they cost individual patients, either through their insurance or individually, about $50,000 a year. The Rituxan is about $60,000 a year. The Herceptin, down towards the bottom, is $70,000 a year. The Avastin at the bottom is $130,000 a year. Patients can't afford these, and insurance companies place barriers so they don't have to pay for them. So they will tell a rheumatoid arthritis patient, no, you use aspirin, or you use a non-steroidal anti-inflammatory drug, and you keep using it. And they just keep putting barriers in the way of using the anti-TNFs. So our goal is to try to bring out copies of these biologics to be able to sell them at a lower price so we can have an impact on patient access. And so you see the biopharmaceuticals' blessings and curse. I've already mentioned on the blessing side, you have molecules that have really, truly revolutionized the treatment of many diseases. 7 out of 10 of the top-selling drugs right now are biologics. Top-selling meaning they make the most money. Not that they treat the most people. They are sold for a higher price. Most of industry, about 30 to 40% of industry that is developing new drugs are developing biologics because they can sell them at a higher price and make more of a profit. And it's also the fastest growing pharmaceutical segment. Fewer and fewer patients are treated by biologics, but costing more and more money. So you may treat 2% of the patients with 50% of the cost of the biologics. And this is mentioned here. It's a major strain on healthcare budgets. We have patient access restrictions because of it. And we have limited competition because you patent the ability to bind a certain target, and then there's not a lot of biologics that are made around that. It's just generally one. So competition is less, so the price is maintained very high. This is a slide that basically shows what you have with rheumatoid arthritis. And if you look at this green box, this is biologic drugs, disease-modifying agents. And you can see that only 50% of patients in Europe, US, and Japan, so these are not underdeveloped countries, only 50% of severe patients have access to a biologic. Severe patients already have the joint deformities that are there. What you really need to focus on is this column. Only 30% of moderate patients have a biologic. If you were to treat all moderate patients, they would not advance to severe because you actually treat the disease. You prevent the joint destruction. So this is a huge patient access issue, and it's something that could be addressed if you could create a biologic that you could sell for less money. This is an example where we have launched a drug in Europe. So we've already made biosimilars in Europe and launched them. This drug is called GCSF, or filgrastim. It's a white cell stimulator. It's like a hormone for white blood cells. White blood cells you make to fight off infection. When individuals are treated for cancer with chemotherapy, a side effect of many of the strong chemotherapies is that it wipes out your white cells. If it wipes out the white blood cells, you're at risk for infection. And prior to this drug being invented, patients would die because they got chemotherapy, could not mount a response to infection, and got an infection. In Europe, this is European data, you can see that in 2009, when we launched a biosimilar to this drug, that we had a steady impact on the number of pre-filled syringes being used. So this is a marker of how much the drug is being used. Because this is expensive, what physicians were doing and healthcare systems is they were using the drug only after patients dropped their white cell count and got an infection. It was designed to be used as what's called primary prophylaxis. When you get the chemotherapy, the next day you get this drug to help bring up white blood cells. If you wait until the white cells fall and you get an infection, it's almost too late to have an impact. But because of the cost, many physicians in many countries were using it in what's called secondary prophylaxis, only afterwards.
Drugs that people might be familiar with, like would Neupogen be one of them?
Yeah, so Neupogen is this drug from a brand name.
Okay.
So Neupogen or Neulasta are the drugs here in the U.S. So Neupogen is this particular drug. Neulasta is a longer-acting form of the same drug. And the combined drugs make about $4 billion a year. So again, very expensive to utilize. And my point here is because it was expensive, patients weren't being treated appropriately. And in 2008, only about 6 million prefilled syringes were sold in Europe. With the onset of biosimilars, there was no real difference in cancer treatment in terms of chemotherapy, but at the same time, far more of this GCSF, Neupogen, our drug is called Zarzio, were used. So you can see we're almost at 8 million prefilled syringes in about 3 years period of time, and this continues to go up. So 30% more patients were being treated because we brought this product in at a lower price. And the general price here was about a 50% reduction. So 30% more patients, 50% reduction. Economies are paying less for better treatment, and more patients are being treated. So this is the reality of what our vision is with one molecule, and we want to do that with others. Other questions? With me? Okay. Cancer has been the number one scourge of humanity, not just in highly developed countries. In Africa, Asia, Latin America, 60% of new cancer cases occur there, and 70% of the deaths. Cancer has become the leading cause of death in emerging markets. Additionally, in the U.S., when you have cancer, patients with cancer have about a 3-fold incidence of going bankrupt. Why is that? Because they have to pay a portion of the treatments that they're being exposed to. And this is a picture of my mother and her great-granddaughter, my granddaughter. And my mother has ovarian cancer. And when she was diagnosed, I worked to get her into a clinical trial that used one of these biologics, Avastin. So she was treated with chemotherapy and Avastin. But like with all ovarian cancer, it recurred. And when it recurred, she was then off of the clinical protocol. Avastin is actually approved for use in ovarian cancer, but not approved in recurrent metastatic ovarian cancer, which is what she had. So the insurance company called her and said, if you want to be treated with Avastin, you have to pay, and it'll cost you $11,000 per infusion. And the infusion happens every 3 weeks. So $11,000 every 3 weeks is what she would have to pay to be treated with Avastin. That's why cancer patients have a much higher risk of bankruptcy. And it's really tragic that this happens, and it's because this agent costs so much, $130,000 approximately for a course of treatment. So that gives you an idea that this is a real thing, happens here, and it's something that should be addressed, and that's what we're attempting to do. So what are biologics and biosimilars? So I mentioned a little bit. Biologics, again, this is a graphic that displays an aspirin, the size of an aspirin here, and the size of a monoclonal antibody here. It's about 1,000 times bigger than an aspirin. And with an aspirin or a small molecule like the drug someone would take for high blood pressure or elevated cholesterol or pain or other types of things, a non-steroidal anti-inflammatory drug, etc., they're made through chemical synthesis. You have maybe an 11 or 15-step process where you chemically synthesize the drug. And if you repeat that process, you get an exact copy of that drug. And when you make a generic to that drug, you can show chemically that your generic copy is identical to the originator copy. And so from a regulatory perspective, when you show that your drug is identical, all you have to do clinically is when you put that drug into a pill form, it's called the formulation. When you formulate it into a pill form, you have to show that when a patient takes that pill form of that drug, that the pharmacokinetics, the appearance of that drug in the blood, is the same. So if the drug is the same, and you show that yours appears in the blood to the same extent, area under the curve as the originator, then yours is essentially a copy of that and can be successfully licensed as a generic. With these, in terms of the biologics, you can make biologics in bacteria and yeast, and you do that for smaller peptides like filgrastim or growth hormone. So this is Neupogen, this is genotropin, for example. And you do the same thing. Recombinant technology, put it into these organisms, and you are able to create a amino acid or a protein. When you go into mammalian cells and you create a monoclonal antibody, the monoclonal antibody is a glycoprotein. So it has not only the amino acid structure, the protein structure, but it has what are called post-translational modifications. It happens after the RNA translates into the DNA, into the amino acids, creates the protein. You get this post-translational modifications. You add on glycosylations, sugars, and other types of things. Those changes are not programmed by the DNA. So we can't program the exact molecule. When we make a copy of it, we can make an exact copy of the protein, but it takes a lot of work to match up these ornamentations, the sugars that are added to this. And that's the challenge with a biosimilar or a biologic. The other thing with a biologic is they're never the same. When you create like Avastin, what my mother was on, it's a monoclonal antibody. When you make one batch, it has certain glycosylations. When you make a second batch, it has slightly different glycosylations. Even within the same batch, you've got a distribution of species of the molecule with slightly different glycosylations. So the originator doesn't even make the identical monoclonal antibody. There is variation, normal variation. And that's acceptable. It's done in our own human bodies. When we make a monoclonal antibody, it's not exactly the same. When we make erythropoietin in our own kidneys, for example, for producing red blood cells, it's not exactly the same. There are different glycosylations that we make. So this is understood. And because of that variability, we can look at it, and when we develop a copy, we can use that as information as to how to best construct a copy of that. So when you create a biologic, you then have the host cells using recombinant technology to make it. You then engineer the process. You grow the cells under controlled conditions. You then harvest the cells. You extract the protein that you want, the monoclonal antibody. You purify it. That is your active ingredient. And then you put it into a formulation, a vial, a pre-filled syringe, so that you can then inject that. So this is the process of making a biologic. Very complex, takes a lot of time. It is expensive, but it doesn't justify the $130,000 you're charged for Avastin. So that's the process of making a biologic. The product you get out at the end has variability. There's slight variability in that product itself. They are not exactly the same molecule to molecule. Now, when we make a biosimilar, I alluded to this, and I use this example. We can make an exact copy of the protein. And if you think of the protein as a Christmas tree, we can make the exact copy of the Christmas tree. What happens is that after the protein is made, you have these additions to the protein. And basically, I look at them as being ornaments. And we all create a Christmas tree. We put ornaments on it differently. Same Christmas tree, different ornaments. We have to look at the ornaments on the tree analytically. So there are very sophisticated analytic tools that we use to look at the ornaments, the glycosylations, the changes that are put on these proteins. And then you have to evaluate those and try to copy them. Now, the originator does the same thing. And I have an example here of glycosylations on a product this was the product before it went through a manufacturing chain. So originators, if they're successful, the drug sells well, they have to make more of it, they do a manufacturing change. They have to scale up the process to make more of the monoclonal antibody. To do that, it changes these glycosylations. And if you look closely, you can see that this peak is much higher on the G2F than this one. And you can see that this little peak here is much lower than the post-manufacturing change. So you can see that these ornaments are different just in making a manufacturing change. Regulators have to look at this and evaluate whether the post-manufacturing change product is the same as the pre-manufacturing change product. And when I use the word same, I'm not saying identical. Same means it has essentially the same clinical effect, efficacy and safety. And this is what manufacturers have to do looking at these ornaments whenever they make a change in the manufacturing process. So there's a regulatory science to this, and essentially it's making the Christmas tree with the ornaments. And when you look at a biosimilar, what Christian Snyder, who's the head of the European Biosimilar Working Party for regulators, says is that the active substance of a biosimilar and its reference medicine is essentially the same biological substance, though there may be minor differences due to the complex nature of production methods. Like the reference medicine, like the original medicine, the biosimilar has a degree of natural variability. So you have to understand this as you're making it. So you don't make an exact identical copy, but the originator isn't an exact identical copy of itself. And this slide is from Christian Snyder also. It shows the number of manufacturing changes that each of these biologics went through over time. And you can see the oldest product is Remicade. It went through 35 different manufacturing changes. So 35 times the manufacturer had to go to FDA or to the European Medicines Association and say this tree is the same as this tree and these ornaments are the same as this ornament. There's a process to doing that. Scientifically challenging, but it's a regulatory process to do that, and that's what's required when you do a manufacturing change. And I'm telling you all of this because of the biosimilar pathway. What we do is we look at the molecules because biosimilar development takes about 8 years. Over that period of time, we're looking at the originator product throughout that time period to see how it changes, how it varies. And that we use that variance in the originator product to form our target about how to make a copy of it. And then once we make the copy, we take it into clinical development, which I'll talk about now. And this slide gives you two pyramids. And it's to give you an idea that biosimilar development is totally different than novel drug development. So if you look on the left-hand side, this is novel drug development. You're developing the drug for the first time. So you can see when you make the product down here in the analytical and non-clinical, all of these data, the analytical, non-clinical, are just to describe what the product is. For Herceptin, it binds HER2. For Rituxan, it's an anti-CD20, by CD20. So you prove what its mechanism is and you show what it is and what it looks like. But the bulk of the file, when you file for a new drug, is the clinical trials that prove which disease it works in, how does it work, and what's the safety profile. And these are very expensive, take a lot of time, and you need 2 phase 3 trials, the final terminal trials. You need 2 of those in every indication to gain approval from a regulatory authority. Now, if you're developing a biosimilar, it's a totally different story. What you're doing is you're doing the bulk of your work down here to show that your molecule is as close to the originator as the originator is to itself, considering these manufacturing changes. Remember, the originator is not the same drug now as it was 10 years ago. So if we say our drug is as close to the originator as the originator is to itself, then it's basically essentially the same drug substance. If it's the same drug substance, then your clinical trial program is very tailored. And all you're trying to do is address any unanswered questions, what's called residual uncertainty. That's usually around immunogenicity. Do you have an immune response to this biosimilar that wasn't in the originator? And so your clinical trials are much smaller. and focused. So a totally different paradigm change. And clinicians, pharmacists, even regulators had to learn this and still have to learn it. Questions? Yes?
The companies that manufacture the monoclonal antibodies, is there a difference in quality of those companies that they can reduce the modifications that occur during the manufacturing process? Like company A is better than company B, or?
Sure, there are differences in your capability. I spent a lot of time with Amgen, so Amgen's well known in this area. They're very experienced in making biologics. Pfizer bought Wyeth, many years ago because Wyeth was experienced in biologics. And so there are things that you can learn over time, and it's very difficult in terms of starting out new to do this. It takes a lot of experience. It's like a practice that you have to learn in this because you're manufacturing with living cells. So obviously, the more experience you have, the better with this. In terms of manufacturing changes, there's not really a good way of controlling it. And companies don't do a manufacturing change lightly because they know they have to go through this regulatory process. In fact, they try to avoid it. But sometimes it's unavoidable because they need to make more product. And so there's not a way that you can get around it. In developing biosimilars, because we learn so much about the process of modifying the process to get a specific glycosylation or a specific ornament on the tree, we actually learn quite a bit more than an originator does because they're not trying to copy another one and change the process to make something look a certain way. What we have to do is we have to make the product, we have to make it functional, and then we have to make it look like the other one in terms of these ornaments. And sometimes the ornaments, most of the times the ornaments have nothing to do with the efficacy. It's just ornaments. Sometimes it does, and when it does, you have to really tune in on that. But we've been asked at times to make changes in our product, even though that specific glycosylation, that ornament, or it's well known that it has no clinical relevance. But because it's a biosimilar, they said, well, make it more like the originator. So a lot of work to do. Okay. Other questions?
I've got a quick one. You talked about the kind of variability that you have to be aware of, that each batch might be a little bit different. Do patients, is that clinically significant? Are there situations where patients experiencing a recurrence might have responded well before and then are up against variability?
It's a great question. So with these changes, particularly manufacturing changes, batch-to-batch variability is minor, and it's controlled by what are called release specifications. So the company has to develop specifications that every batch has to meet before it's released. Usually those specifications are around the mechanism of action, how that drug works, or binding or something of that nature. And if you vary outside of a fairly narrow release specification, then you as a company can't release that batch. And that's to protect patients, to make sure they have the same effect. The point here is that patients should not notice any difference whatsoever between drugs over time, even though there's multiple changes, because of the process that's involved in controlling and convincing regulators that it's the same drug. And same thing with biosimilars. Because there is that flexibility, you can then show that it has the same efficacy. Now, some changes, some manufacturing changes that happen are fairly dramatic. And then a regulator doesn't know. And one example is Aranesp, which is a longer-acting erythropoietin. And they went from a roller bottle manufacturing. So you take a bottle, you seed the cells in it, coats it on the glass, you plug it up, put media in it, it rolls through the media, produces the product, drain it out. Very archaic way of doing it. And when they scaled up and they were selling more of it, they went to a bioprocessing, a big vat that they did. And to do that, they had to change the master cell line. So a totally different cell line. Because of that, it changed the glycosylations quite a bit. The regulators looked at that and said, I can't say that this molecule is the same as that molecule, so you have to do new clinical trials. So they had to then repeat clinical trials to show that their molecule was the same. Once they completed those clinical trials, phase 1, phase 3, and a 1,000-patient phase 4 study, then the regulator said, okay, I'm convinced it's the same molecule. Even though it's not the same molecule, it has the same efficacy and safety. For a biosimilar, that's kind of what we have to do. Even if we convince them that the molecule is the same analytically, by law, we have to do the phase 1 and the phase 3 trial. Yep.
Higher efficacy than other ones. Are there any efforts to repeat the same kind of glycosylations that batch had? Is that possible?
You would never. The challenge is that a clinical trial, to show that your product is better, you use statistics to do that, and you have to be statistically significantly better to say it's actually better. And the clinical trial is very non-sensitive to pick up minor differences. You have to do a big trial, or you have to have a molecule that has a big difference in it to pick up differences. And so you would never experience that. And we get that a lot because there's an anti-biosimilar campaign. All the companies that are making these products, that make the $100 billion, they come up with all sorts of rationale and barriers about why we shouldn't license biosimilars. And one of them is, well, if it's more efficacious, then it must not be as safe. And how do you prove that? So you would never, even in a batch-to-batch variability, you would not see a statistically significant clinical response. But people do learn by that. So one example is rituximab. And Rituxan basically is an anti-CD20 antibody. It binds to CD20 on B cells, which is a white blood cell. And you have B-cell tumors, like B-cell lymphomas. And you want Rituxan to bind on that white cell, that malignant white cell. And because it's an antibody, it has this Fc part of the antibody that sticks out, and it attracts a natural killer cell, your own immune system. So your own immune system recognizes that Fc portion of the monoclonal antibody binding to the malignant white cell, and the natural killer cell kills the cell. Now, we can change the ability of that Fc portion to attract a natural killer cell by glycosylations. And that was found in doing these types of work. And so a company would then improve that ability to attract a natural killer cell to kill the white cell, but in doing so, it would be a new molecule. So what companies have done is they've tried to improve Rituxan over time, and they've improved what's called this antibody-directed cellular cytotoxicity, ADCC, the killing. But what happens is when they improve that killing, they worsen the safety profile because it causes side effects and it binds to other things and kills other things. And so by making a little change, it may improve the efficacy, but it also may worsen the safety. Okay. So on a biosimilar, this is just a simple diagram to say the aim of biosimilar development is not to reestablish safety and efficacy or benefit to the patient. It's to establish similarity of the reference compound. You've already shown that the compound is the same analytically. Here's clinical data that helps confirm that similarity. And this is not understood by physicians. And the companies that make these products really try to confuse physicians about this. So they will say, the biosimilar isn't identical to our molecule. And what does that say? First, it's biosimilar, which really means biodifferent. It's a lousy term. And then someone says, by the way, that biosimilar isn't identical to our compound. That seeds doubt and fear. What they don't say is our compound isn't identical to our compound, which is the truth. So they say a partial truth to stimulate fear in terms of what biosimilars are. And we have to remind regulators also about this. When we first started meeting with regulators 6 years ago or something, and I remember this specifically. This is John Jenkins from FDA, and this is a nice quote. He said, I think we probably were in the mindset of we always had to have comparative clinical trial in biosimilars. And that was the case. You show up with this big package showing the molecules are the same. The clinician has the mindset that this is whatever. It's a filgrastim, a Neupogen. Well, if you're going to be developing a filgrastim or a Neupogen, here's the 3 big clinical trials you have to do to show that it's safe and effective. And they had to step back and say, no, that's not the question. The question is, how do we confirm similarity? And it's a different mindset, different approach. And FDA's had to learn that as well over time. And it took a lot of meeting with FDA and cajoling to get them to that point. And this is an example where in Europe, in the US, Australia, Japan, New Zealand, they have these specific biosimilar regulations where you have to show that your product is what's called comparable to the originator. And you can see here, this is essentially a gel, isoelectric focusing gel, with on one lane is the originator, on the other lane is the biosimilar. And they're exactly the same on the gels. Here is the same product on the outer edges, and in the middle are all of these follow-on biologics that are supposed to be copies, but they're made in Thailand and Asia and India and others, and they don't have a regulation that says it has to be comparable to the originator. So a lot of people call these products biosimilars, but they're not. We call them non-comparable biologics. Now, it doesn't mean they won't work. They can work. They can have efficacy. But what it does mean is that they're not comparable to the originator, and you shouldn't be able to extrapolate to information about the originator unless you prove that it's the same. This is a graph as well that restates the similar finding differently. This is our GCSF in Europe, our Neupogen. You can see the black line up here, which is Neupogen, and then the other originator that's up there as well, which is Shugai's product. So both of these were originator drugs on the market. The entire market was only these two originators. Biosimilars came on the market, and you can see how Sandoz came on, and then we've taken out. And right now, we're the number one Neupogen, the number one GCSF in all of Europe, by volume, not by cost. Amgen still makes more money than we do because they sell it for a much higher price. But by volume, we're the number one in Europe. If you see this red line, this is all the biosimilars together, because there's several biosimilars. And if you add this line, which is as high as the original one, with what's still being sold by the originators, this is where you get your 30% more volume. So it just shows that we're able to take on the originators, have more volume, more patients benefit. So key takeaways from this: biologics are expensive, access is limited. We've got many, many examples of that. And you probably, well, you're too young, but you might know someone in the near future who needs these medications and has difficulties. We've got $10 to $15 billion of biosimilars that are off patent now. And then we also have about $100 million that will be off patent in the near future. Developing biosimilars already improve patient access. I mentioned that with our GCSF, and save money in Europe. They're already stimulating a biologic revolution worldwide. More people are getting treated the way they should be treated earlier to prevent the disease from becoming very severe. And in reality, it's the right thing to do at the right time. Although we still have a lot of challenges, because if you're going to save that money, someone is going to try to prevent you from doing that through a variety of ways. And even now, in Pennsylvania, there's legislation that's being proposed to help with this situation. And what happens is, eventually we will have what are called interchangeable biosimilars. That means if the physician writes for Humira, which is $50,000, and we have an interchangeable biosimilar to Humira, and we're selling it for, let's say, $20, still expensive because it takes a lot to develop these, the pharmacist can give you the lower priced one, so you don't have to pay a co-payment of, I don't know, $5,000 on this. And to do that, the pharmacist needs the legal authority to make that change, just like they do with small molecules. That's what a pharmacist does with generics. They have the authority to do that. They don't have the authority to do that with the biologics because there's not a law for that. And because of that, we need legislation. And we've tried to propose legislation that makes the most sense. Others are proposing legislation that is more preventive. You have to do these 50 steps before the pharmacist can make that change because it will be a barrier to its use. So even in that situation, there are barriers that we're running into. So with that, I'll close, and hopefully you're awake, and we'll ask any other questions. Thanks for your time. Jeff?
You mentioned early on that the next wave is gene-based therapies. When do you foresee that we'll start to see those things come to the market where people can actually take advantage of them?
Well, gene-based therapy, it can come in a variety of ways. So things are happening right now. University of Pennsylvania is doing some work where, from an ex vivo perspective, they're taking lymphocytes out of a patient. They're using ex vivo gene therapy to upregulate a specific gene in those cells, and then they're infusing them back in so that they attack a specific malignant cell. So it's in the research phase. 2 companies are proceeding with that to try to bring it through as a approved product. But when you take those cells out, when you have to modify the gene structure, when you use recombinant technology to insert a gene that may not be there, it's very, very expensive. So we're talking $100,000 here for some of these drugs. This is going to be $600,000-$700,000 for a patient to do these types of things. So what would be the natural progression of that? It would be, can we do it in vivo? Can you somehow use the gene therapy in vivo and not go through all of the manipulations where you take the cells out and have to manufacture all of these other biologics to influence that cell? So we've got a ways to go on that. But I mean, it's promising, but there are many hurdles as well with that new technology. Yeah.
What is the relative, shelf life of these biosimilars, since they are proteins and susceptible to denaturation? And would there be any dangerous side effects of having a biosimilar that is old?
Yeah. Well, it wouldn't be biosimilar. Let me put it this way. I'll talk about biologics, because biosimilars are a biologic. And what I don't want to do is say, well, there's a shelf life issue with biosimilars that isn't with biologics. All biologics are the same, and they do have shelf lives. And with these, you basically have to maintain them what's called a cold chain. So they have to be held between 2 and 8 degrees. Okay? So when you make them, they have to be put in 2 to 8 degrees. When you ship them, they have to be within 2 to 8 degrees. When you store them, they have to be within 2 to 8 degrees. If you keep them within 2 to 8 degrees, their shelf life is between 2 and 3 years. If you take them out, if they heat up, if they do others, then the protein can degrade, etc. What generally happens in this type of situation, if you have a monoclonal antibody, let's say that was shipped and somehow the cold chain was lost, and they set on a dock at 40 degrees centigrade. By the way, these are all centigrade, so 2 to 8 centigrade. If it sits at 40 degrees centigrade, 90 degrees, 100 degrees Fahrenheit, that will increase the liability of the molecule itself. If you were to infuse that, generally speaking, what you do is you lose efficacy. You don't cause a problem with like monoclonal antibodies. There are rare instances, and EPO is one, erythropoietin is one. So we make erythropoietin on our own body. All of these other monoclonal antibodies we don't make. I mean, they're provided exogenously. We make erythropoietin in our own body, controlled by our own genes and made by the kidneys. When you have renal failure, your kidneys fail, they stop making erythropoietin. If you have a problem with erythropoietin, unstable, it forms aggregates, you can develop antibodies to the aggregate of erythropoietin. If you develop antibodies to the erythropoietin you give exogenously, you develop antibodies to your own endogenously produced erythropoietin. And that produces a condition called pure red cell aplasia, meaning that you can no longer produce red blood cells. And then you have to be supported by blood transfusions. So that is a rare thing that can happen if you don't make the erythropoietin correctly, if you don't store it correctly, if you don't do that. And that happens. And there was a major issue that came out when Johnson & Johnson was making this in about 1992. You usually get one case of PRCA in 10,000 patients. And that's because PRCA happens spontaneously as well. They had about 100 cases of PRCA in a very short period of time, and it was related to a change they made in their prefilled syringe. And they caused leachates from the prefilled syringe, which then causes aggregation of the protein, which then caused immunogenicity. So these are the things that gets back to the question of how good you are in manufacturing. It really takes a lot of understanding of how you manufacture, how you control, how you ship. But this is for all biologics, and biosimilars have to be treated in the same way. Any other questions?
These are so expensive to do, you can't, from a marketing, a business standpoint, how do you pick which ones to do? So many years before the other patent is done, or how do you pick and choose that from a business standpoint?
It's a great question. So what happens is you look at how easy it is to do, what's the patent life on it, and what's the relative value. It's very simple for the ones that we're working on because they're $8 or $10 billion drugs a year. So if you just even capture a little bit of that, it makes sense. But you do have to plan it 8, 10 years ahead, and that's a problem. You also have to look at these, the originator and how it varies over time. If you just start out right now and you have no information, no data on Humira, for example, you don't know what the variability is, you don't have a very good history in terms of what is your target to hit. So you have to think way in advance in terms of what you're doing with these things. And it's also very costly. And as companies go through various things, you plan for the future. So we're investing now for the future. But it's a very expensive time for us, for example, to invest. And we haven't sold these products in the U.S. yet because the regulatory process wasn't there. So right now it's a very difficult time because we're investing all this money in the research and we're not selling the products yet because they're not on the market. So you have to have a mindset of investing for the future. It's almost like if you start your own company, you use a venture capitalist, you go out and you raise a lot of money, and you focus on this one product, and ultimately you're successful, you pay back the money. But there's a risk associated with that. The company has to deal with that risk.
Avid Biosciences.
Biosciences?
Avid.
Avid. Yes, I can't remember the technology.
There's one.
From any different technology?
That's what I know so far.
Yeah, there are many different. You can produce monoclonal antibodies in tobacco, for example. So in Kentucky, they're producing monoclonal antibodies. This Z-Mab that's being used for Ebola is being produced in tobacco plants, for example. And then now they're wondering how do they scale it up.
Going through that phase 3 process, a drug called bevacizumab
That was it? Yeah.
and Cotera.
Yeah.
Those two. That's where they're.
Am I getting the chain?
One more question, and then we'll wrap
Is
this
there?
up.
Do you utilize Six Sigma a lot with biosimilars more than you do with generics?
Six Sigma for
biosimilars, are companies utilizing?
Yeah, you do that, and we have Six Sigma black belts that are involved in terms of narrowing down the variability through all of this. You have quality by design to really focus on these types of things. One of the challenges anytime you use those types of tools is you have to have a process that's very well controlled. And you start out with a process that isn't as well controlled, and then you narrow down over time to really controlling your process. But yeah, they do. And for me, it was interesting. I think I was sharing with Nan previously. I came from California in the Bay Area to Germany, and I'm kind of a rebel at heart, an entrepreneur. I need boundaries that are there. And I go to Germany, and Germany and Austria, they're very process related, controlled, et cetera. And you'd think that'd be a lousy mix, but as it turns out, it was really quite good because they needed a little bit of entrepreneurism. I needed a little bit of chaining around me, so the combination worked out pretty well.
I want to thank you so much, Mark, for being here and for giving this very interesting talk. Mark has been here much of this week because last night he was one of about 20 alumni from across the university, including the campuses, who received the Alumni Fellows Award, which is the highest honor that the Alumni Association bestows on an alumnus or an alumnus. So we were incredibly proud to be there with him when he received the award. He received this wonderful kind of sculpture of Old Main that's very heavy, and he has to fly back to Germany. Then we gave him a framed poster of the elms outside of Henderson. Again, he has to fly back to Germany. But we have a tradition of giving our distinguished alumni speakers a framed poster from the advertising that we did for the presentation. We promise we will send all of these gifts to you in the mail so that you don't have to lug them on your flight. But we thank you very, very much for coming this week, for coming to classes, for meeting with our students, and for kind of giving us the thrill of getting to sponsor you at the Alumni Fellows Dinner. It was really fun. So please, let's give him one more round of applause.
Thanks, Nan. We were walking down. I told my daughter, because I saw the poster out there, I said, we should steal that. So now we have one.
You could. We probably have a few others we could give you, but you have a framed one. And there's a little reception, just some snacks out there, and please feel free to come down, and if you have additional questions for Dr. McCamish, you're welcome. So thank you for your very good questions, too. We've appreciated that.